All right, looks like we've got the bulk of our registered attendees logged in, so thank you so much for joining us. If you're just joining us now, my name is Kelsey Gibson. I'm going to be your host for today's webinar session. So if you experience any technical difficulties during this time, feel free to chat me in the chat panel of your GoToWebinar interface or reach out to me in email at kelseyg@labkey.com. Today, Ryan Luce, our product manager for LabKey Biologics, is going to be covering the core areas of the platform and going into a little bit of a live demo of what LabKey Biologics looks like and what it's all about. So if you have any questions during the session today, there's a question panel in GoToWebinar. Go ahead and submit those throughout the presentation. We'll take them at the end, and I'll also do a little bit of a pause for anyone who has questions that come up at the end of the session there. We are also recording this webinar, so if there's anyone who you would like to share the recording with, I will send that out after today's session. You should expect that email tomorrow from me, the recorded version. So without any further ado, I'm going to pass it off to Ryan Luce. Take it away.
All right, thank you very much, Kelsey, and thank you all for attending and taking time out of your busy day to learn a little bit more about LabKey and LabKey Biologics. So I'm just going to reiterate one thing that Kelsey said is, as you have questions during the course of the webinar, go ahead and send those over, even though, and then we'll make sure to get to those questions at the end. We leave plenty of time for that. So, all right, so we're going to go ahead and go forward here. So real quick, the agenda for today's webinar, first of all, just a little bit of kind of the evolution of LabKey, current challenges in terms of managing this type of data with spreadsheet-based systems, what is LabKey Biologics, core components, and then we're going to make sure to demo, at least in a light manner, some of those components within the application. Questions and answers, and then at the end we'll tell you how to easily sign up for a free trial. This is online hosted, so you can play with the application yourself. So kind of walk through from there. So first, just a little bit about the history of LabKey. LabKey has really been around for over 15 years now. This is our 16th year. We started in 2003. Developed to support proteomics data management at Fred Hutch. And really, we were spun out of as our own standalone company in 2005. And between 2003 and 2015, you know, we've got just a couple of points here, but you can imagine the literally hundreds of different features and projects we did on behalf of academic research institutions, biotechs, clinical trial management companies, et cetera, to extend the LabKey platform to make it better able to manage diverse sets of biomedical research data. Really starting in 2015, LabKey kind of started to accelerate its development. A big part of that was our work with the 100,000 Genomes Project, which is a National Health Service project to have data on 100,000 participants there. We had a public portal around the Zika virus outbreak. And then most importantly for this audience, in 2017, we launched the LabKey Biologics application to support in concert, developed this with a company here in Seattle called JustJust Bio Therapeutics. I've had a number of other clients come on over the last 2 years, and we've really made a lot of headway in building an application that helps support protein engineering and general sort of R&D activities in molecular biology. So we've been around for a while. What were the challenges that we were trying to address? And so there's a couple of things just to kind of focus on current systems for managing data. And we focused here on spreadsheet-based workflows. So some of those challenges are that we get poor quality and inconsistent data about biological entities. It amazes me the number of times people store sequence information in Excel, for instance, and some of the formatting changes that are formatting challenges that can create and inconsistent data results. This can be extraordinarily time consuming. It's very error prone when you're trying to do manual integration of data from various different data sets. There's a lack of visibility into prior research. When you store information in Excel or store information in PowerPoint files, those are not easily queryable and searchable from kind of a central data repository framework. And then ultimately, this is a loss of productivity. It takes longer to do science because of those inefficient workflows and data handoffs. So these are the challenges that LabKey was determined to address in the development of LabKey Biologics. So how do we deliver on that promise? So first and foremost, at the top left, we have an easy system, easy registration of biological entities, and I'll show you some examples of that. So registering of protein and DNA sequences, cell lines, antibodies, whatever the different sort of important building blocks for you are, the system is configurable to represent that. There's a direct connection between experimental data and sequence information as a result. By bringing in that particular assay information, we can see that data right there in the system. Much of LabKey Biologics is built on the foundation of tracking the lineage of samples. So that can be just a couple of generations of sample derivation, or one of our clients, for instance, has 14 different generations. Because we've tracked the lineage of those samples, we can go back to see its original context and see what cell line was used, what molecule has been created, what media recipe has been used. It's important not only to kind of facilitate tracking that lineage, but to also make it queryable in a performant manner. And data, but that a user can retrieve it fairly quickly. This LabKey is a customizable system with the flexibility to grow. So many of our clients have, upwards of 30 to 50 and even 75 different assay designs as they're bringing in different types of assay data into the system. We have tools to manage laboratory workflows. Ultimately, protein engineering, molecular biology, all of these types of R&D activities, this is team-based science. And so you need mechanisms to keep everybody on the same page, to pass work from one to another, understand what others are doing. LabKey Biologics has mechanisms to do that. And then finally, and this really gets into I hesitate to say this, but the DNA of LabKey is integration with external software and instrumentation. LabKey has always, even prior to LabKey Biologics, made it very, very easy to get data into the system, whether that is manual manipulation and bulk upload, or accessing any of the many, many APIs that we've generated, so you can both insert data into the system as well as pull it out. So those are kind of the general, the philosophy, if you will, the things that we are serving up with the LabKey Biologics product. So what are those key application components? And I really want to focus on these 4 highest level areas within LabKey Biologics. There are some other sort of tangential pieces that we can talk about as well, but these are the 4 key things. So the first and foremost is entity registration, registering molecules, sequence, and cell lines. Tracking samples in terms of what is the definition of those samples, where were they derived from that. Then taking assay data, tracking it back to the samples from which it was derived. So you've always got kind of that persistent connection between the two. And then defining experiments. What is the set of samples and assay data, files, and results that we want to tie to a particular group of samples within the system? So let's walk through each of those. So first and foremost is entity registration. Now, entity always seems like kind of a not descriptive enough word for what we're talking about here. These are the building blocks of your molecular biology. So those can be protein sequences, DNA sequences, RNA sequences, cell lines, antibodies, things of that nature. And we maintain relationships between all of those various entities. So if you have registered a particular line and a vector or construct, what is the molecule it creates? Or once you do make a sort of full-length molecule, if the signal peptide is cleaved, what are the new mature sequences that are generated? All of those relationships are kept within the system. And working in the background, we've developed an automated classification engine. So when you register your new heavy chain, we automatically classify the regions and features within that based on the classification engine. Calculate molecular weight, make sure that we understand other physical characteristics of those sequences. We also make sure that that sequence is unique. So if you are re-registering a protein sequence or DNA sequence, obviously the system makes sure that you don't do that and add in additional information to previously registered pieces. We also make sure that the ID for this is always unique, so that you've got a unique identifier that you can use both within LabKey Biologics, but also in adjacent systems to keep track of all of those entities that are important to you. Let me take a little. So now I want to show you what that looks like within the system, actually. So I'm going to go over to my browser here. So by the way, and LabKey Biologics is a web-based system, meaning that you access it via the browser generally. Now that can be both hosted by us, or if you want to, you can install that on-prem. And we have got a heady mixture of both of those scenarios within the system. So this is when you log into LabKey Biologics, kind of the screen you land on immediately. You can see we've got a focus on a search bar, and then a number of those areas that I was talking about, some of which I'll give more data on going forward. So on the registry piece, you can see we have all of the different entities within the system that we've configured for this particular piece. So in this case, I really want to focus on the molecules, protein sequences, and expression systems. So if I go to, for instance, molecules, now many of these are actually antibodies that I've registered. So it's a mixture of multiple protein sequences. We've configured the system, built the system in a way so that's a very flexible option for you. So you can register as many things as you want. And this is all the molecules in the system. In this case, I've got just 13 or so, but that can be in the hundreds or thousands within the particular system. And this grid is fairly searchable. If I want to go find each of the proteins that, molecules that has PS20 in it, it will easily filter down to that. I can filter different pieces of this. The isoelectric point, all of those different things are available to you. So I'm going to remove that filter and go navigate to one of these particular molecules, in this case, M3. Which is, I'll probably butcher the pronunciation here, but glembatumumab. So this is an IgG2. You can see we have information about its mass and other physical characteristics of this particular antibody. And we have a little bit of information about the light chain and heavy chain that are within it. So in that case, I can drop that down and actually even see more information about PS13 in this case, or PS14. All of these things also are links to be able to navigate to get even a more detailed look at those particular entities. In this case, we have built an expression system that will generate that particular molecule. And you can see we still have links on this detail view of the expression system as well. We know that it uses CHOK1, it uses these particular constructs and these particular vectors and generates M3. Ultimately, also, we can see information about the samples that have been generated from this particular expression system. So these are downstream samples based on this expression system design. These are physical vials that are related to this particular expression system. I also just want to mention, going back to our molecule, if I go to PS13, for instance, we're storing a lot of information at the sequence level in that classification engine piece. When I registered this sequence, it identified likely leader sequences, regions, and features within the system. And so we can kind of keep track of that, the annotations there. You can add and subtract annotations if you want to. And if you're really interested in, CDR2, for instance, I can select that. You can see it's a really fairly short sequence of amino acids up here. And if I want to, I can copy that little subsequence, and it's now on my clipboard to take into whatever tool that you want to use for building your next molecule. All right. So let's go back to our PowerPoint. So that gives you kind of a sense of the relationship between all those various entities. And I hope you get a sense of that you're always kind of walking between them. All of those things are links, so they can always get more context about a particular sequence or cell line or whatever it is that you're looking at. All right. So that's entity registration. What about sample tracking? So I kind of mentioned this. You're able to connect. So when you say oh, this is my expression system parent, and derive 3 different bioreactor runs that are built on that expression system, follow that by purification, whatever sort of sample derivations, you can trace the lineage of those samples through many, many generations as you need to. You can then also view the aggregated assay results for all of the test runs against a particular sample. So back in the application, let's take a look at that. In this case, these are particular samples within the system. And you can see for each, we have 2 different sample types. We have expression system samples, and then just a generic bucket that we call samples. You can create as many sample types as you would like with different fields for each of those. Maybe you want to store project X. Maybe you want to store the location. Things of that notion are very easily customizable and configurable within the system. If I go look at a particular sample, and actually I'm going to just go look at one. I'm going to search for a particular sample. So this sample detail, we don't have a lot of fields on this particular sample. If I need to, I can edit things within them. But one of the interesting aspects is that lineage piece, is what does the lineage of this particular sample look like? And you can see here that this sample comes from expression system 3.1 and then went through a couple of different generations to ultimately generate the current sample that we're looking at here here that's highlighted. Now, sometimes these types of graphical interfaces for exploring lineage can get really overwhelming. If you have more than 10, or maybe you have 100 different sample IDs, it gets really hard to kind of navigate that within this graphical view. And so we also generated a grid view of these lineages as well that you can access via this link. In this case, I want to actually see the parents of that particular sample, and we can see that particular view. Now, maybe I want to actually see all the children of the parent of this particular thing. And you can see now we've got this is an example where there's lots and lots of children associated with a particular sample. If I actually look at that sample detail, you'll kind of see what that looked like in graphical view. And that is a good example of why we felt like we needed to build a grid view of this as well. So back in our kind of slide here, we've talked about entities, samples being generated from those entities, and we have really easy mechanisms for generating new sample IDs in a way that makes sense to you. If you want the parent to be a part of that, if you want the date, whatever it wants to be, we can make sure that your sample ID makes sense and also is unique within the system so that you don't accidentally duplicate that. So the next piece is being able to look at that assay data. So one of the nice things about LabKey is that by having a consistent assay design, so you have assay designs for each of the types of data that you want to collect, it enforces a consistent data structure and that allows for querying as well as analysis of the data. You can view those results side by side with the sample, other aspects of the entities connected to it, and lineage information. And I'll show an example of that. And then there's easy manipulation and analysis of results using both tools within the system, as well as if you need to export this into other analytics tools. Remember I said it's very easy to get data in and out of LabKey. Sorry, I keep going a little bit hoarse here. I'm drinking some water. All right, so let's go take a look at those examples as well. So let's go. We don't have a lot of assays in the system. I've only built 3 different assay designs. Like I said, many of our customers have 40 or 50 or 75 of those. And so you can have a much more interesting heat map, if you will, on activity within the system from this particular view. But if I go look at, for instance, our amino acids assay design and go examine the results within, you can see we've got a sample ID, and this is a link out to that particular sample. Sample date, description, which we've left blank here, and then all of the 20 different amino acids fields that are connected with these particular samples. And so each assay design can be a little bit different. If I go and examine, for instance, SEC, you can see that that grid is very different. We still have sample ID, but we have a number of different fields that we've connected to those particular samples in this case. One of the nice things, though, about that, for instance, on the amino acids, is we can also generate within the system grid views of that particular data that we want to access on a routine basis. So in this case, I built a view that not only includes the sample ID, but what is the expression system that generated it? And then I'm just focusing on 2 of the columns, in this case, So if you look at that, you can see we now have our sample ID, the expression system parent that we pulled in via its lineage, and just those 2 pieces of data. And then if I want to go look at, I only care about expression system 1, we filter all of that data down to just those samples. So we have custom grid views available to you, and you build those within the admin interface. We also have charts of this particular data. So in this case, I've built a chart that plots these sorted by the expression system. And so as you add data into the system, you don't need to rebuild this chart. Each time, this chart will basically take whatever the filters are on this grid and build it anew for you. And you can see that we've color-coded these by the expression system which they're connected with. All right. So kind of the one last area I want to make sure to demo in this webinar is management of experiments. So one of the pieces of feedback we got in the early days is, although we had great mechanisms for tracking the lineage of samples, connecting it to biological entities and assay data, is that that sometimes it felt like the data was not partitioned off. There wasn't a mechanism to partition things off into a single experiment, which is obviously how scientists generally think about doing this. They don't want to just think about adding data to a big database. They're doing a particular experiment. So we built this new concept really over the last year to group all the relevant sample and assay information by experiments. This allows us to automatically view all the assay results for a defined set of samples. So once you say what your samples are, it shows the assay data for that particular experiment. You can also attach related files to an experiment within the system. So I'm just going to give a quick example of that. So for instance, if I've just generated a couple of samples, let's do some sort of a selection of these. Let's say we're just looking for those particular samples. I'm going to select them all, and I'm going to add those to an experiment. This is going to be a new experiment. We'll call this webinar demo. And it's just as easy as that. So we're created a particular experiment. We can view that experiment, see those samples that are there, and I think you can see how responsive the system is. Now, if there's any assay data connected to these samples, and I'm not sure there is, we will see yes, there is some assay data in this particular case. You've got SEC data, we've got titer data, and amino acid data already into the system. If we add more data tied to those particular samples, more of that, all of that data will automatically show up within this particular view. And we can go look at these things within the system as well. Now, one kind of nice thing is I'm back on that amino acids grid. I still have those grid views available to me. And in fact, now I can actually build my chart that I built previously, but it's just tied to the data that is in this particular experiment. We're really trying to kind of make that process of analysis more efficient. If you are constantly making the same grid views or constantly making the same charts, making sure that that's sort of programmatically available to you wherever you're looking at that data. Okay. So those are kind of the four, at a very high level. You know, these types of demos could go hours, which I'm sure is not what anybody wants to do with their time. But I also just want to mention a couple of other areas that are helpful tools within LabKey Biologics. So we have built a module around media registration, so we can create standard media recipes, defining, you know, the ingredients that are in a particular recipe as well as what their concentrations are. And then each batch of that created, you can connect those lots to other downstream data as data is generated. You can even make batches that are slightly modified. You have a particular recipe, but in this instance, you want to change the target concentration for one of its ingredients, or you want to add an entire ingredient. You can still use that same recipe and make a modified batch. Also kind of tangential to the four key areas I showed you is workflow management. There is a request system for requesting analytical data or other tasks. You can group sets of these tasks. So this is for a particular, you know, we're going to analyze the results of a single bioreactor run. I have created requests for many different assays. I want to keep those grouped. And then there's a persistent connection between the request itself and the resulting data. So it's a project management tool that sits within the application that sort of helps enable that team-based science, but that connection is persistent. So I can go see why we did a particular experiment later if we need to. So those are some of the other pieces within there. And I kind of want to, before we, I want to mention one of our JustJust Bio Therapeutics. And this is a quote from their Vice President of Molecular Design, Randy Ketchum. You know, the comprehensive utilization of LabKey Biologics at Just Bio Therapeutics has allowed our scientists to have a consistent system to store and retrieve entities and data, enabling increased capacity. Ultimately, we're trying to help accelerate insight, ask those questions and get them answered more quickly to facilitate that next discovery. So in general, working with LabKey is different. We are designed for science, but we are built on a flexible platform, trying to reflect, you know, the different ways and different workflows that different laboratories may have. Building out your own set of entities, building out your own specific assay designs. We know differences exist, but it all with kind of that science foundation. We do incremental implementation, meaning we don't sit back and take a year to get from you know, to get this system up and running in your situation. We try to do incremental. How can we deliver value as quickly as possible? Our development cycle is very focused on being innovative and agility, and I'm sorry, innovative and agile. So we follow an agile development methodology, making sure to get those highest priority next features out to our users as quickly as possible. And we believe in lasting partnerships. Many of our clients have worked with us for over 5, if not 10 years now, across the board of a variety of different LabKey implementations. We are really bought in to your long-term success. So The other thing to mention here is that you can go sign up for LabKey Biologics with a free trial right now if you want to. This is a hosted version of the system. You get it for 30 days free. There are sample data, so you can play with some of these aspects, building your own entities, building your own samples, putting in assay data into the system, and start to see how that works in an actual environment where you get to use it directly. So I'm just a couple of minutes over my 30-minute goal, but in terms of presentation, so I want to open it up now. What questions do people have about the system? We can dive back into specific areas or general aspects of it. So open it up to questions.
All right, thanks, Brian. Yeah, if you have any questions, feel free to put them in the question panel there. I know we had a couple of audio cutouts there, and I apologize for, but so if you have any clarifying questions, please ask those. We've got one question that just came in asking about whether or not biologics is free. Ryan, you want to talk a little bit about the licensing model?
Yeah, biologics is not free. It is an annual subscription. Although one thing I would mention, and we can do that both hosted or for you. In terms of the licensing piece, if you discontinue using biologics, you lose a lot of that UI and functionality. But you can still kind of down migrate into the LabKey Community Edition, so you will have all of your data. It's a little gentler downgrade path than a lot of options, we think, but LabKey Biologics on its own is not free. Now, the caveat to that is you can sign up for a free trial so you can play with it directly and really have a working system, but that's just kind of to get a better understanding of how it might work for your scenario.
Okay, great, thank you. We have another question about what happens when you import a GenBank file.
Okay. I have an example of that actually. So let's go back. Let's just do that. So let's say you have developed elsewhere or on the web or something like that, a GenBank file that represents a plasmid that encodes for the molecule that you're going to create. So in that case, for that particular entity, we can import, you can see there's a variety of different sort of file types here, including GenBank files. So let's go find a particular file. Demo files. Nope. Yeah, this is a good one. So in this case, it took that, I should have shown, it was just a standard GenBank file with a particular sequence, and it is for the cloning vector PZCAA. But it not only created the construct, but it also created the protein sequences that encode for it. In that case, these different sort of resistance genes for that particular plasmid. And we've got kind of those persistent connections. If I go look at that particular thing, the sequence is available to us right here. So all of it not only kind of created the construct, but it also created associated with it as well. One thing I didn't mention on all of, I kind of alluded to it, this is sort of tangential to the question, is we have mechanisms, if I want to register, for instance, a protein sequence within the system, we can do this through our wizard here, or we can, and so just kind of see what that looks like, we can either translate from an existing nucleotide within the system, or we can just manually add a sequence, whatever we want to do in those two particular cases. And that's sort of a one-by-one experience. Generally, though, oftentimes people, wait, I want to import a whole bunch of data. And so we can allow you to do that in Excel or FASTA files or a CSV file, any variety of different file types to get data into the system. And we actually even provide here a template for what that Excel file, for instance, needs to look like. But we also have APIs. So we have a couple of clients who have separate systems for designing their protein sequences and then use the API to register those entities into LabKey Biologics. So for any piece of data you see here, whether it's the registry, the samples, the assay data, anything, there are APIs available so you can connect instruments or connect other pieces of software to pull that data into the system.
Great. Another question here. So this one is kind of a two-parter. Does everything start with an entity? This user is a genomics core lab and everything is project or experiment based. How customizable is LabKey for overall workflow?
Yeah, so we definitely have, it does not have to necessarily start with an entity, although we also might create entities to represent that specific workflow. This configuration has these entities, but you can add additional entities, and we would work with you in that situation to do that. It's very straightforward. We would just want to make sure it's set up appropriately. But it doesn't have to be. You know, these workflows can start from a sample or other aspects as well. I'd want to get in a little bit more into the specifics, but I'm very confident we'd be able to kind of address the specific needs of your scenario.
Okay, great. Thank you. See if anyone else has any questions. Go ahead and add those now. I'm going to give folks just a minute or two more, and we'll get those answered. If not, if you think of a question after the session, I will be sending out the recording. Feel free to send any questions to me, and I'll make sure that those get to the correct person to get to the answer that you're looking for as well. So conversation doesn't have to stop here. We're definitely available to talk a little bit in more detail about your specific scenario, and definitely do take a moment to go to look at the biologics trial. It's a great environment to explore the platform on your own and at your own pace, and generate an idea of your potential use case and how this would work for your environment.
And Kelsey, we'll make sure to send this link directly out to the participants here, right?
Yes, absolutely. I will do that.
Okay.
Yeah. All right. Well, it doesn't look like we have anything else. So thank you, Ryan, so much for walking us through the platform. And thank you, everyone, for taking time out of your day.
Thank you very much.